My name's Summer Johnson. I go to the University of Vermont. I'm majoring in data science and minoring in computer science and mathematics. I'm originally from New Jersey, but I've been living in Vermont right now. So I was working with Carlos Garza on this project. We're working underneath Dr. Garg, and what we're researching is Chagas disease. We're looking for different protein biomarkers and mitochondrial DNA biomarkers that can be used to either diagnose an individual with Chagas disease or find out if they have acute or chronic Chagas disease, because at the moment we aren't able to determine that. So what we did some background research into different work that's been done with cardiomyopathy, different cardiovascular diseases, and we found different proteins that the levels of which have differed in blood samples. And what Dr. Gard decided to do was she had a human-led trial. It had 42 individuals, 12 of which were healthy individuals, 15 had Chagas disease but were asymptomatic, and 15 had Chagas and were symptomatic. And what we did was we took the levels of each of these proteins, as you can see here, in both their plasma and serum, which is their blood, as well as these different mitochondrial DNA types. We took the levels of those, and we performed a number of different statistical methods to find out if there was a significant difference between individuals who are asymptomatic and symptomatic, or individuals who did or did not have Chagas disease. So we used binary logistic regression, single predictors with these two binary outcomes. From that we found the predicted probabilities. We developed some ROC curves, and from those ROC curves we found the area underneath the curves, which is what we were able to interpret in the end. And as you can see graphed here, it's a bit more interpretable in our radar plot, which has the averages for each symptom group. But we have PARP 1 and HNRNPA1 levels. It's insignificant for being able to predict if someone is symptomatic or asymptomatic if they have Chagas disease, but it is you can see it's above this line, it is a very good predictor for whether or not someone has Chagas disease. So most importantly, from what we pulled from this, these are our two diagnostic biomarkers within somebody's blood type. So hopefully this will be able to be used in the future with vaccine development, and being able to take blood samples from just any individual and find out if they have been infected by the T. cruzi parasite and will develop Chagas disease later in life, so that we can better treat them now, even though they don't have symptoms. I had a really good time. I don't have a big biostatistics background. It's been primarily data science. So it's been nice being able to work with people who, this is their life work. Dr. Gard's been working with Chagas disease for forever. So it's been interesting to learn about that, being able to hone in some of my statistical skills and being able to apply it to a more biological sense. I've really enjoyed that, as well as working on a team, being able to work in a lab with a bunch of people, and then the program itself, I've met a lot of people, other students who are like-minded, and I get to learn from them.